Hello, my name is Rugan Suresh and I'm a ninth grader at Piney School in Osprey, Florida. The name of my project was Ability of Microalga Chlorella vulgaris to remove nitrogen and phosphorus from wastewater microgravity. The inspiration for this project was something we can all relate to: water. It's something we can't live without. In fact, it's not just people, any living organisms need water to actually survive. With this, as human society is advancing, we need many innovative and sustainable solutions to help increase water availability for everyone. One of these methods is water recycling and treatment, where contaminated wastewater or urine can be cleansed and taken out its contaminants and pollutants out of it, so it could be used. One emerging green solution in this is microalgae, which use specific compounds such as specific pollutants such as nitrogen and phosphorus, which are present in the wastewater in its own food supply basically, and in turn also produce a byproduct of its algal biomass, which can be put into biofuels or antioxidants or any pharmaceuticals. And with this, as water is needed not only on Earth but in space, which is humans' next frontier, we could use microalgae to help improve things in the ISS, as water recovery system, or in life support systems as we go into deep space explorations. In my school, we had this opportunity to actually test this out in this program called SSEP, Student Space Flight Experiments Program. In this, we were able to submit a proposal with this idea and try to get a chance to actually perform our experiments in the ISS. It was a long shot, but with help from many mentors such as Professor Ralph from the University of Technology in Sydney, also Professor John Kilbane, and also Professor Serena Ergas and Dr. Philippidis and their whole team in USF, we were able to get a whole proposal together and an experiment design to all get ready for the actual flight. And in the end, our team got picked to actually be sent from the Kennedy Space Center, which in November of 2023 actually launched and returned back in January in 2024. With that, before we could actually even send it up and finalize the experiment design, we had to perform many trials to analyze how the algae could best be grown in microgravity. Even though previous research has been done with microalgae, especially even with the strain Chlorella vulgaris in the ISS, we have found that specifically it's best to be grown heterotrophically, since in the ISS we weren't sure of a guarantee of exposure to sunlight. So we decided to use a carbon source of sodium acetate along with a dilution of synthetic urine, which would be the wastewater that would be decontaminated by the microalgae. And the dilution we used was a 1:25 ratio. After the whole proposal and flight and everything and return had occurred, we had to get ready for the post-flight analysis. With that, we had 5 main things we had to do: the total nitrogen, total phosphorus, the COD, which is the chemical oxygen demand, the lipid concentration, and the total suspended solids. All of which we tried to collect after the flight to see how the microalga Chlorella vulgaris spent its time in the ISS and reacted with the wastewater. We had quite a few limitations in in the project and designing it, especially since we had to confine our whole experiment into 8 milliliters, which was in this exact actually tube, the FME kit, which went around the ISS about 642 times. Because there was such a small space, we had to perform our experiment on a very small scale, but still keep the ratio of like how much wastewater to algae was used in the experiment. We also had a third volume in our FME kit, which was a glutaraldehyde fixative, just to end the experiment to avoid any error or skewing of data while the microalgae was leaving the ISS and returning back to Earth. After this, and analyzing everything, which we also had only one shot to do, which we had to make sure everything was done perfectly and with utmost care to make sure we get the proper results and data that we were needing to, because we only had one shot to do something like this in maybe our lives. After analyzing all our data, we came to the results that the total nitrogen utilization was higher in the ISS samples compared to the ground experiments, which was very surprising. That we were hoping that at least it would be similar so that it could be used on the ISS or in space, but somehow the algae managed to even outperform itself than on Earth. Total phosphorus and total suspended solids and chemical oxygen demand was similar in both the ISS and the ground truth experiments, while also lipid content was higher in the ISS sample, which could have meant that the algae was able to grow more and take in the nitrogen and phosphorus better as a food source in microgravity. With this, we started to need to take this data and decided to want to share this and have some future plans with this. One main thing we've been doing is trying to go to many research conferences and share our findings to other people in the scientific community and our peers as well, just to get some feedback and just see how we can improve on this project and take the next big steps for this. When this is finalized with different strains and different conditions, we could start this could be starting to be put into a larger scale in the ISS veggie labs or on the deep space exploration a life support system to make sure that we can travel as far as we can, in addition to everything else that we're using to keep our water available no matter where we go. This whole experiment, on a personal level too, has allowed me to see a new topic that I haven't I've always been interested in water sustainability, but I never I started to research about microalgae and its uses too, only with this project, which has been very fascinating to understand how something that causes red tide could also be used in things that could actually help all of human society. I would also really like to give a big thanks to our teacher facilitator, Mrs. Rosen, who is the 6th grade Earth and Space Science teacher at our school. She has led this SCB program at Pineview and helped us so much and supported us so much for our project and experiment and everything through this process. She is not only a great inspiration to me, she's also a great inspiration to many students. Also, I would like to thank you for taking your time to watch this presentation. Thank you.